Welcome back to part 2 of Unit 3, Lesson 2. This part of the video is optional unless you've missed class. And we're going to continue talking about why does the body perform replication. But first, we want to make sure we're very clear on what replication is. So replication is a process of making an identical copy. In other words, a replica. That's where replication comes from. Of all of the DNA within the cell. And you might recall way back when, Science 9, that this occurs during interphase. So why does the cell go to all the effort of making an identical copy of its DNA? Okay, its purpose of making the identical copy, the simple answer is you need 2 copies of DNA for when the cell divides. So that each new cell gets a complete and identical copy of the blueprints for the entire organism. Okay, great. So it needs 2 copies for cell division. Simplest answer. But why do cells divide? So why do we make copies of the cell? Why do they divide? There's many reasons. The first and perhaps the most obvious is cells divide for the purpose of growth. They can only get so big themselves. As they get bigger, they divide so that you can grow. Now, if you're a bit of a klutz like me, or maybe decide to play contact sports, you also need cells to divide for the purpose of replacing damaged cells. And of course, it's not just being a klutz or that. Your cells just get damaged over time due to day-to-day use, so it replaces damaged or worn out cells. And cells also divide in a slightly more complicated process to prepare gametes. Your gametes are the cells for reproduction, so we're talking sperm in males and oocytes. That's a cool word, oocytes. It means eggs in females. So those are the key reasons cells divide, and in order to divide, they have to make copies of their DNA, and that is done by the process of replication. So now we're going to look at how replication occurs. And first of all, we have to know it's DNA to DNA, and because we don't want to damage the DNA, we want to keep it in a nice safe place, it must occur in the nucleus. Now what I'd like you to do is just roughly divide your page into quarters. No, actually fifths. So go down your page about halfway down, and I'm going to have to guess about what's halfway down on mine. So go about halfway down your page, draw a line, then on the top half divide that into 3 even sections. And those who had me last year, this is going to be very familiar because we don't really have to increase our details or knowledge from last year, but we have to make sure everyone's on the same page and that you actually remember what you learned last year. So we're going to have 3 panels on the top, and then we're going to take the bottom half and just divide that in half. So we have 5 panels all together on our page. And for those who write rather large, I'm going to challenge you to write a little bit smaller today. So replication occurs in the nucleus. You start out with entire set of DNA. And of course, I'm just going to draw one little strand of DNA, not the entire set, but this applies to all of the DNA that sits in the nucleus. And what you're going to have is your traditional double helix. Okay, and in that double helix, we would call that the parent. Oops, sorry. The parent or template or original strand. Okay. And then to replicate, our first step, well, we've got that DNA and it's all coiled up into its chromosome form. We have to start by unwinding that double helix. And we do this using an enzyme. I'm going to color my enzymes green just because that's how I learned them at university. An enzyme, I'm just going to make a little circle here, called topo. That's really its nickname. Its full name, it's topoisomerase. So what happens in step 1? Topoisomerase unwinds the DNA double helix. Now, it's called topoisomerase. That seems like a really difficult name, but topo refers to topography, which means it's 3D shape. Think of topographical maps showing hills and valleys and all of that. Iso, well, think of isosceles triangle. Has 2 sides that are that's right, same. Iso means same. Isomers are things that are chemically the same but have a change in their 3D shape. So nothing's changed chemically about the DNA, it just went from being wound to unwound. Its 3D shape has changed, but its chemistry is the same. So the iso refers to the chemistry being the same. And anything that ends in ase typically indicates that it is an enzyme that helps speed up a reaction. Helps speed biological reaction. Okay, so now that we've got the first step, the next step, we've got it unwound, we have to essentially unzip it, break those hydrogen bonds. So what happens? See that? This enzyme that's called helicase I kind of draw it like a wedge shape because I view it coming up and wrecking it, tearing it apart. So you still have topo going along, doing its job, moving up the DNA, and following in behind it is helicase. And it's typically said unzips the hydrogen bonds between the bases. Didn't plan that very well. Essentially separating the two strands. So if we looked at something and we had A, T, G, C on this side, then the complementary would have been T, A, C, G. And the bonds that were holding them together are now broken thanks to helicase. Okay, I'm just going to stop there because I'm afraid I'm going to run out of time in the middle of the next panel, and we'll pick it up in the next part of the video.